Our first speaker will be Anda Trifan, at the University of Illinois at Urbana-Champaign. Her field is theoretical and computational biophysics. Her advisor is Ahmad Turkorsan, and her practicum was at Argonne National Lab.
Thank you. I'm super excited to be here. DOE CSGF is extremely important to me and so special, and I'm glad that they let me come back here. I was supposed to give this talk last year, but ironically, I got COVID. So like 2 or 3 days before my flight, I had to test, and I had COVID, so I couldn't come. But anyway, I remember getting the phone call that I received the fellowship and really literally jumping up and down for joy, but I don't think I really truly understood the impact it was going to have on my life. And I'm going to go through my scientific talk maybe a little bit faster because I want to also touch on my personal experience as well. And this is very different from what everybody else's expertise is in this audience. I do molecular dynamics and biophysics. So I'm going to touch on 2 different projects that were winners, and one of them was a winner for Gordon Bell, and then the other one was a finalist. So SARS-CoV-2, which is the virus responsible for COVID-19, has affected all of our lives, and it still has, and it caused millions of deaths around the world. Upon the entry of our bodies, the virus first makes contact with the ACE2 human receptor, and it initiates the fusion of our cell's membrane with the virus membrane, and it allows the virus to release its genetic material into our cells. And then a human ribosome is hijacked, and all of this mRNA is being translated for the parts that are needed to replicate the virus within our bodies. So this is called the replication transcription complex. This is a complex of proteins that together are able to replicate the viral RNA in order to make new viruses constantly and release them into our bodies. And while we have a lot of experimental data in the form of cryo-EM and crystal structures, these are often difficult to decode, and also they're just static images. So we have absolutely no real information about what conformational changes undergo in these proteins. So we can use molecular dynamics, which is often called a computational microscope, to give some temporal resolution to these structures. But there are 2 limitations of molecular dynamics. One is the time scale that you can simulate for. This is very limited. It's on the femtoscale time. And also the size of the system that you are able to simulate. So the more atoms you have in a system, the less time you're able to simulate, because these calculations are extremely computationally intensive. So because of this, we can use enhanced sampling techniques like weighted ensemble simulations and combine them with AI or ML to learn more in a shorter period of time. So like I mentioned before, this is the ACE2. I don't know if okay. This is the ACE2 human receptor, and this is the receptor binding domain of the SARS-CoV-2 virus, and it sits on the spike protein. I think everybody has heard about the spike protein by now. And in order for this binding to occur, the receptor binding domain has to undergo very large conformational changes. It goes from a flat conformation to open, so it can reach the ACE2 receptor. And this type of very large conformation is not feasible for just regular equilibrium molecular dynamics. And this is why we used enhanced sampling techniques. So the weighted ensemble simulations were done in Lilian Chong's lab in Pittsburgh, and she provided terabytes of these trajectories, which we used as training data for Deep Drive MD, which is a workflow or a pipeline that's generated in Arvind Ramanathan's lab at Argonne National Lab. And we use these very large sets of simulations to train models to generate these embedding spaces at lower dimensions, and then we extract some of the physically the more rare events that occur in the simulations to use as novel starting points for simulations. And so we tend to propagate just the more rare conformations and then sample more of the conformational space that is available to us. So this is an example of a low-dimensional representation of the embedding space using the T-distributed stochastic neighbor embedding, or TISNE. And we use just a standard method for finding outliers or rare events. And here, the larger spheres in the TISNE plot correspond to a specific conformation of the spike bound to ACE2. So we are able to physically extract different conformations from this analysis. And with this methodology, we're able to actually find conformations that are also experimentally determined. So we were able to drive the bending of the knee of the spike, which is a necessary conformational change in order for the spike to bind to the ACE2 receptor, to 46 degrees, which is actually very close to what we find experimentally. So here is the spike. Here's the transparent helices are the initial conformation, and the solid red is what we find. So with Deep Drive MD, we were actually able to effectively speed up the simulations by about 8.3 times. And also, we observed about 25% more conformations in only 12% of the time. For even more massive complexes, such as the transcription complex, which is a very large protein, you can see these are real-time cryo-EM structures, and there are very fuzzy areas, which means that we have very poor resolution, or we cannot really understand these areas. So we can use molecular dynamics or atomistic simulations and combine them with fluctuating finite element analysis, which allows us to have these same systems, but on much larger timescale simulations. And so we used all-atom molecular dynamics to equilibrate and to simulate these structures, and then extract important information such as van der Waals and interactions between these protein-protein complexes. And then we fed this information to the FFEA simulations, which were performed by Sarah Harris's group in UK. And they are able to do these simulations that give us a lot more information on the whole complex conformations. And on this type of time scale, we're able to observe also the binding and unbinding of different domains. So this is a representation of the same protein with FFEA mesh. So if you look closely, these are different energies that are obtained from molecular dynamic simulations to kind of guide or give parameters to FFEA. And this is an example of an FFEA simulation, and you can see that there's a lot more movement than you would have in all-atom molecular dynamics. So one other example of what we can use molecular dynamics atomistic simulations for is RNA unwinding. So the unwinding of RNA within the replication transcription complex is a very important part of the replication mechanism of the virus. And so this is actually about, for like this 5-second movie, it's about 3.5 months of work where we had to do a lot of different parameterization of non-equilibrium methods, and it was, let's just say, a lot of work. But by combining some of these non-equilibrium methods, by pulling on the sides of the RNA, because we have information about the active sites in the replication transcription complex, but we pull on this RNA in a very specific way because you have to maintain the helicity of the RNA in the complex. So we use extra bonds to keep these base pairs together, and then we have kind of a rail for the RNA to move through. So this is a force that keeps the RNA inside, and then pulling to get to the active site. And so this was one of the first kind of simulations showing the unwinding of RNA and the importance of the different conformational changes that undergo in the replication transcription complex. So from all of these different scales, we're able to see with FFEA very large conformational changes between the two different complexes. So this is NSP 1014, and this is the RTC, or replication transcription complex. But we see movement between these two domains. And then, although we also see movement with the all atom simulations, we observe a lot more movement at the atomic level, which is to be expected. Yeah. But we don't see the intramolecular movement as much as we do. So why are we doing all of this? All of this work is very relevant because although we have a lot of information on structure, we don't have a lot of information on dynamics. And so we have had a lot of efforts directed towards vaccines and towards the prevention of infection, but obviously a lot of people still get infected. So we definitely need methods to address the post-infection mechanism of the virus. And here's a really nice movie. Here's the ACE2 receptor. This was actually done by NVIDIA for our Gordon Bell presentation. Here is the ACE2 receptor. And one of the problems that we've had over the past 2 years with the spike and just focusing on vaccines and focusing on treatment that allow for prevention is that the spike has mutated several times. So that's a big issue when you are developing something because you don't know how those mutations will affect the medicine that you're making. So in order to kind of bypass this, all of these proteins that make up the replication transcription complex and the proteins that are translated from the genomic RNA are severely conserved. So they're about 96 or 98% conserved from previous SARS-CoV viruses. So this would be a much more effective way to treat the virus. So obviously, we would want to prevent, but if we can't prevent, then we want to treat. So yeah. I've been just incredibly fortunate to work with amazing people. And these are my advisors. Ahmad is my advisor at UIUC. Arvind is my advisor at Argonne. And then Romy was part of this Gordon Bell work. And when I said I wanted to talk a little bit about my personal life is I see a lot of women here today, and I'm extremely excited about that because I feel like although it has gotten much better in the past years, women in our field or science in general are not as common as they should be. So I was very fortunate to get this fellowship, and during my grad school, I was at a point in my life where I was ready to have kids, which is kind of rare for most people. And DOE was just incredibly accommodating for that. And I'm extremely grateful for being able to get all the aspects of my life together. So for the Gordon Bell talk, actually, I was 7 months pregnant, and I had to record my part of the talk. And thank God we had a lot of really good people to do post-processing sound, because I ran out of breath an incredible amount of times because of that. So these are just a few of my awards and papers just to show that you can have kids, or you can pursue the things that you want to pursue in life and do the things that make you happy in your professional life. So you don't have to choose, and it's not kids necessarily. It's like if you want to hike or whatever, do do something. Yeah, but you can do both. Like there are ways to compartmentalize your life in such a way to feel like a well-rounded individual. So yeah. Questions? I get the yellow.